Okay, thank you. So I will talk today about scalable microbiome representation and specifically going beyond just taxonomic and functional annotation. So first, when we talk about representation, what we mean is when we take a microbe, of course, we can have DNA sequences of that microbe, or we can have protein sequences of that microbe, or anything else. But in the end, what we're going to end up with is some kind of feature matrix where we have features in the columns. And this is what I call representation. And we can then feed that into a model to perform tasks. Now, why we are even interested in that, in representing microbes in general, I think we have heard already today about some of these topics. But for us in our lab, we are, for example, interested a microbe adapt to a specific environment or to a specific plant phenotype. So what genes, what proteins, what functions, and we would want to understand that and also be able to predict that. Now, there are some problems with the current microbial representations. So I think the most common way right now is to do whole genome sequencing or even high-quality metagenome sequencing, and then obtain protein-coding genes. And then from the protein sequences, there are obtain functional annotations. I think there are many that you probably know, and we have also heard talks about this. But I think there are several problems with these functional annotations, and one of the main ones is that even for a couple hundred microbes, we still obtain thousands of features, and this is quite problematic for the AI models and for the performance and generalizability. And also, they are quite slow to obtain. So for us, what we were interested in, comparing the traditional functional annotations to protein language model embeddings, and specifically in this step, so when we translate the protein sequencing into annotations. And we were specifically interested in the runtime coverage, so how many proteins we can annotate, number of features, and performance. Now, what were we evaluating on? So we had around 1,000 microbial isolates that were whole genome sequence. We obtained representations, and then the task that we were interested in was presence or absence on the root of a plant a few weeks after inoculation. So when we talk about prediction, so this step, we compared two models, multi-layer perceptron with a random forest. And then when we talk about the input, we compared two functional representation. One is the KEH database, which I will call KOs from there. And then we compared to simple protein families obtained through orthology clustering, which I will call OGs. And then we have the protein language model embeddings. If you know something about the protein language models or the ESM2, you know we don't really obtain microbial representation from this. So I want to explain a bit how we got there. So if you know a bit about this, you will obtain from a protein language model or ESM2 amino acid level embeddings, but it is a common practice to then pull, so we can, for example, take an average across the amino acid and obtain a protein embedding. And we can follow a similar logic when it comes to the microbiome level. So one microbe has also several protein embeddings, and we can pull them to obtain a microbe level embedding. And what I mean when I say pooling, we can again take a mean, like is done in the protein level, or we can use attention pooling, which is a bit smarter way to do that. Now I want to present some results. So this is for the annotation part. When we look at the KEH, so KOs, we can see that we can annotate around 50% of the proteins, and we obtain thousands of features, and it still takes quite a long time to run. When we look at the protein families, which I call OGs, we can annotate all the proteins, but we really obtain a lot of features, which is not very feasible to then train with, and it also takes an insane amount of time to run. So when we compare that to the ESM2 embeddings, we can annotate all the proteins, and we only obtain a couple hundred features, and it also only takes around 2 days to run, and this was also just done on one thread, and it can easily be parallelized on multiple threads, so it can be done much faster, actually, than this. Then I want to quickly get onto the performance on the task, which you can see on the right there. So when you look at the models that work with the KEH inputs, you can see that across all the metrics, they obtain quite high performance, around 0.8 or higher. When we look at the models that work with the protein families or OGs, they do obtain quite random performance, as this is a lot of features for the model. to work with, which is slightly improved, but still not that good when we apply some basic filtering. Then when we work with the ESM2 embeddings as an input to the model, we can also see that we obtain quite high performance across the matrix of around 0.8 or higher. It is quite important to know that this was evaluated on a kind of a random test set. We have also done more evaluations, so on external data sets or also on what I call hard test set of taxonomies that the model has not seen, but for the sake of time, if you want to see that work, you can follow us. And I want to touch on a different topic that I think is quite popular with the protein large language models for the rest of the talk, and that is interpretability. So when we work with these models, are they interpretable, or is it just a black box? So firstly, we look at the embeddings. So what do they really encode? So one thing that we did was we picked 10 random protein families or OGs. We have done this multiple times, but this is just one plot of that. And you can see that the individual embeddings cluster based on functions. We have also done this on taxonomy, but we were also thinking prediction is stronger than just seeing a plot. So for taxonomy, what we have done as well is we have the microbe embeddings and we calculated similarity, and based on the similarity, we obtain hierarchical clusters that we then compare to the real taxonomy hierarchy. And then we calculated an alignment score on the genus level. This alignment is quite high, so 1 here would be perfect alignment and 0 would be random. When we look at the family, this drops a bit, but still provides quite moderate to strong alignment. So from this, we can see that actually, even without fine-tuning the model, the embeddings do contain some functional and some taxonomic information. And additionally, we can also analyze the features. So if you remember this model architecture, we can check what proteins was the model paying attention to. When it comes to the mean pooling, we can, for example, use SHAP, or we can analyze the attention weights when it comes to the attention pooling. There are other methods, but I just want to mention these two. And with that, I want to leave you with some conclusions. So in our opinion, the functional representation can, of course, work very well, but there are some clear advantages to using protein language model embeddings. For example, the runtime, if you have a lot of data, or the amount of features, if you, on the other hand, have too little data, and the performance is still quite good. And then also, I think it's important to note that these are all quite simple approaches, and they can, I think the performance can still be much improved. So, of course, we can do fine-tuning on the protein large language model. We can use more complex architectures. We can use more complex pooling, which we have seen plenty of talks here at this conference about. And additionally, also, like, we would really like to do work on more tasks for which we need more data. So for us, like, the scalability is really important. And yeah, if you're interested in that, you can follow our work. And with that, I want to thank my group, especially Gijs, who has generated all the data and did all the bioinformatics pipelines that I was working with. And if you want to see and are interested in plants, you definitely should see his presentation. Thank you.
Thank you very much. We have time for a question.
Okay, thanks. I'm here. Just wanted to ask about the evaluation.
Yeah.
What have you done any on it?
What do you mean? You mean the tested evaluation or the?
Yes.
So the evaluation that I showed was a traditional train validation test split, but we have also done evaluation of 6 external data sets, and then evaluation on this kind of like hard test set where it was split. So the test set there were random samples that were kind of easy that the model has seen the taxonomy of, and then 100 that the model has not seen the taxonomy of. So those were kind of like 3 types, but I just showed the test set evaluation, which was like 60-20-20 split.
Thank you again.